Welcome to Sequence the Future. Today, I am excited to be with Jeremy Ellis. Jeremy is the Chief Scientific Officer of BioID Genomics and Fry Laboratories. Jeremy, it's a pleasure. Thank you for joining me.
No problem. It's a pleasure to be here, and I appreciate it.
Absolutely. So, why don't we go ahead and have a blast from the past? Let's dive in a little bit into Jeremy Ellis's childhood. Can you tell us a fun fact about growing up?
Sure, absolutely. So I'm an Arizona native. I was born and raised here. I think something that people find a little bit unexpected in terms of being a scientist is that I was my high school mascot. So
Wow.
yeah, surprisingly enough, I got to be at every football game and every basketball game and be dressed up in the suit. The mascot's name is Toro Man, which is a bull wearing a jersey. So it was very hot and sweaty, but it was a fun entertainment. I got to be around all the cheerleaders and be part of the action. So
That sounds amazing. So the question is did people know that you were the mascot or was it supposed to be a secret?
it was supposed to be a secret. Actually, yes, it was, and it was a little controversial at the time. People would, when I would be amongst crowds, particularly some of the larger schoolmates of mine would try to open up the mouth and figure out who I was. So eventually they did figure out, it was me, but it was for a little while I was able to be anonymous, and people were wondering who's Toro Man. So it was a fun time. Got to throw out a lot of free T-shirts up into the stands and go to a lot of games, travel around. But it was very sweaty, very tiring, and I got to be the first and last pictures on my high school year. So.
That's amazing. I bet you were a legend once people found out that you were the mascot.
Yes and no. But yeah, it was fun. Being a non-cheerleader, going on the cheerleader bus down to different cities and stuff, and hanging out with all the fun action. It was a good time. So yeah.
Yeah, that's so interesting, Jeremy. So tell me a little bit about, I mean, growing up as an Arizona native, tell me the quick bio of how you ended up in the sciences. What are there some childhood dreams that have been realized? Tell me a little bit about that.
Yeah, in a sense, it is. I feel that I was always curious from the very early on. I think I was lucky with my family, where many of our summer vacations were not just relaxation vacations or just entertainment. Many of them were educational in nature. So museums, science museums, learning about nature, exploring nature, going to the beach, and not just being at the beach for the beach, but being at the beach for the biology, to see the birds and the crabs and the seashells. So that sort of instilled a general wonder for science and the natural world. And
Is that because of your parents, Jeremy? What was it, a mother, a father? Who was it that was so interested in integrating, right, you kids in nature?
Both, but I think from a different point of view. I think from my mother's point of view, from an education point of view, being an educator herself and a teacher, she took every opportunity to turn every experience into a learning experience. So that's just who she is. From my father's point of view, is more of a philosophical perspective about the world, of being in place with nature and understanding that the natural world around you is not just grounding as a person, but it's also a powerful tool. If you understand how the world works as a mechanism, then you can be a more effective person in your career and life. So,
That's
but
incredible.
that's sort of where it started. Reading textbooks at a very early age, just trying to understand what were the tinker toys that made reality. Chemistry textbooks when I was young. So it started from there. I really think so, and then just spiraled forward. So. Yeah.
That's so interesting. And so, is that something that as soon as you started going through your official education out of high school into college and beyond, was it immediately known that you wanted to keep going with the sciences?
Yes, I think very early on. It never really varied from science. I think I bounced between being a paleontologist for a little while, a biologist, marine biologist, a molecular biologist, um, geneticist before that was even really a concept precisely. So, it was a very early ideas of studying genetics and molecular biology. So it was never a question of if I were to be a scientist, it was what type, really.
Yeah. How did you hone down on kind of precision medicine, genomics, right? I mean, science is obviously a vast,
Mhm.
vast opportunities within that realm. How specifically genomics?
Um, that's a great question. So I've always been interested in the morphology of organisms, and this gets into my choices of how I arrived at my educational path. So why does our body plan look the way it does? How does that patterning get laid down during development and embryogenesis? Those types of genetic mechanisms and programming were of interest to me personally. Um, and that just naturally gravitated towards molecular biology, genetics. Now, where I land today with genomics and precision medicine, that's really, it's not a plan. It just happens, and that's the way that the world you gravitate towards as you develop a skill set in graduate school. In my case, studying Drosophila. It just so happened that the skills that I developed there are impactful into human health, and that was always a hope and a thing that I wanted to make a difference. I just didn't know how it would, manifest in a way. How could I, help people with being able to understand how their bodies are the way they are? Well, along the way, I picked up these skill sets: informatics, bioinformatics, sequencing, data analysis, things like that.
Got it. So it's almost like you may not have known specifically how things would unfold in your career and what you do, but it just over time, kind of that natural evolution has led you into what you're doing now.
I wouldn't disagree with that at all. I think that's accurate. And I think that finding something that I enjoyed and that happens to be a career was important. So I pursued what made me happy and what I found that was interesting. And a career exists here. So I'm thankful for that.
That's wonderful, man. I think that more people could benefit from that perspective, that if they follow what makes them happy, right, they'd probably be able to make more of an impact and just feel better about their journey themselves. What is it, if you wake up on the wrong side of the bed, and you're just not feeling well, I'm not saying necessarily sick, but maybe you're just not feeling particularly motivated. What is it that is a motivator for you, or your why?
Yeah, that's a really good question. Um, and I do believe that I can persevere through a lot of adversity and differences. And I think that going through graduate school was particularly a challenge for anyone who goes through that type of education. It's a never-ending journey, and you don't have a defined goal. So how do you get there? It's basically perseverance and pursuit of the puzzle. So
Hmm.
and I hold as a personal you know, uh, trait that I try to live up to is persistence. And I find that most problems can be solved by persistence alone, and not giving up or letting that erode your belief that you can overcome some challenges. The pursuit of the puzzle is where you extract the joy from the problems. So, and I view many of those either work or work related or technology related challenges. It's a puzzle to solve. It's like a Rubik's Cube or any other sort of brain teaser puzzle. How can I look at and inspect this problem in a different way that hasn't been thought about before? And this really relates to some of the technologies and innovations that we've developed in the lab. Is they were born out of necessity. How do I analyze 1 million reads to the species level from a DNA sequencer? That would take months on a standard benchtop computer. How can we address that type of a question and extract clinically meaningful data when the canonical wisdom is saying, don't do this, do something else? So, and it's those
Wow.
puzzles that I find interesting and motivating. So.
Oh, that's great. Can you give me a specific example of, a problem that sticks out in your mind that you were able to overcome and some of the lessons learned?
Well, the one that I mentioned is a great example because I think it's easy to understand. So,
Sure.
a DNA sequencer, a next generation DNA sequencing, when they were first released to the wider scientific community,
Yeah.
these are devices that are converting biological data into digital data, and early on there was a very massive gulf in how to analyze that particular data, and many of the research tools would be, for a lack of a better term, bootstrapped together to give you an adequate answer. So in the case of microbial identification, you would sequence a bunch of DNA fragments and then you would group them together based on an arbitrary threshold of homology, saying these are very similar, these are very similar, and these are very similar. And then you would assume that if you were to identify members or a representative of those groups, that those would all be share that same identity. So if this group of reads was Escherichia coli or E. coli, and this one is C. diff, and you'd say, okay, those groups are those organisms. But we arrived very early on that had a high degree of imprecision. And so that challenge of identifying every single individual read in a timely manner where we're not analyzing them for months gives you a lot of interesting innovations. How do I speed up the database comparisons? Is there ways to look at the data at different levels of granularity? Do I need to actually look at very precise base pair differences between these sequences, or can I start breaking them into groups earlier based on their relatedness? And that is a perfect example where we took something that would take many weeks of analysis on a computer
Sure.
to 30 minutes. So, and it's that type of targeted continual process to wheedle down to what's actually necessary to get a result that's robust.
That's incredible. And no doubt over time, as you encounter obstacles, I'm sure that it's increased your confidence having overcome a lot of the problems that you've experienced.
Yes, I do think that's the case. I also think that even if I were to not gain traction, I would still persist. You know, it's one of those innate features. I will try regardless. So, but it does help to have some successes, but to know that, hey, if it doesn't work today, you got tomorrow. If it doesn't work tomorrow, you got the next day, and you keep working on the problem honestly.
Yeah,
so,
absolutely. I'm curious, Jeremy, since you really have had your boots on the ground and direct impact on kind of the evolution of next generation sequencing and genomics, what excites you maybe about the next decade, right? If you're pulling out the crystal ball, like this is where everything's moving, where do you think it's moving? What excites you?
Absolutely. That's a great question, and I wish I had a crystal ball. And again, this is only from my limited viewpoint of the expertise and the sort of the technologies that I have my fingers on. So I think that the first near-term innovation over the next decade that we're going to see is a transfer from clonal omics approaches. So if you're doing DNA sequencing and looking at inferring a DNA read from a population of DNA sequences and then saying that there was a founder molecule that reflects. So all of your main types of sequencers are that way. We're going to see a move to single molecule sequencing. So every individual molecule, so every DNA read itself, the diversity in those reads will be reflected. So I think that's the first new layer. That's going to open up a whole new level of precision with detecting low-level SNPs or heterogeneity in samples that were believed to be homogeneous previously because of technological assumptions. That will have impact in precision medicine, things like inherited diseases, responses to treatments, medications. We're going to see that will be certainly the case in microbiology applications. That's the near term. The longer term, I believe, will be a synthesis of many of these omics-based approaches, genomics, proteomics, transcriptomics. You're going to see a synthesis, I think, of these approaches where you will have unified devices and approaches that will look at the proteomics or the proteins in a particular clinical sample and what the expression is telling you. And that combined data will tell you with more precision about how to treat or diagnose a particular patient with a disease or illness. I think that's going to be the first, and then the tools that will follow. So I think we've already started to see this where bootstrapped, academic-based tools are not going to get us there, and you're going to need consumer-friendly, non-expertise level tools. These are going to be tools that are lowered and will be able to predictively generate meaningful results for people who don't need to be experts. And I think that's going to be the next level. So synthesizing all of those omics, and then a layer of tools, a suite of tools that will be your expert for you and give you the answers you need. I think that will be the next 10 years. So.
That's fascinating. I love the descriptive way that you went about that. And it seems very pragmatic that that's kind of the next steps in natural evolution. Tell me a little bit about if you have a favorite quote, maybe a current philosophy of life that guides you. Is there a quote that you can share with us and why it's your fave?
Yeah, sure, sure. As for philosophy of life, it's really never giving up. That's just the case, because that's what you can control. For quotes, though, I think one of my absolute favorites is from Carl Sagan, and I know that's aiming high. Um, it goes something like, if you wish to make an apple pie from scratch, you must first invent the universe.
Oh,
And I think that it's very impactful because it touches on so many different things that I hold dear to myself, is understanding the mechanisms of reality, chemistry, what the parameters are, what your guidelines are, because DNA only does DNA. It does the things that DNA does, and understanding what it is will allow you to use it in new ways, use the technologies in new ways, recover from errors, develop new technologies.
yeah.
And so all too often, I think that molecular biologists are becoming kit users. They open a box and they run the kit and they get a result and they don't necessarily know what happened. And that's a problem, I think. And I think if you are honest about your scientific curiosity, you should be asking, what is happening at every single one of these steps? What is occurring? I can't tell you how many times I've dissected a kit or a product from a third party, which was mysteriously intractable for some reason, and then understanding, I see how this works. And then you can use it for something completely new. And before you know it, you solved a problem that you didn't even know you needed to fix.
Wow.
Yeah, so I think that that's a really apt quote. So just be a student of all sciences, and then you can invent apple pie. So.
That's wonderful. I love that. And I think you demonstrate that just in our brief conversation here, that you seem like you're really a student of life. You realize that there's kind of an ongoing, endless education that happens.
Yeah, I think the only other thing to say that to talk about is I have consulted for several science fiction authors
No
on different
kidding.
books. Yeah, absolutely. New York Times bestseller on a couple of different books, and it's a little surreal seeing your name in a book in in Barnes and Nobles or some bookstore somewhere. Your name is in an acknowledgement for being a science consultant. But yeah, they're taking it seriously, putting guardrails on the science fiction, making it real. That way people can learn and be excited by it. That's something I hold dear. So yeah, I hope to inspire in that way.
That's incredible. Are you able to drop what the title is where we can see the work that you've been a part of?
Yeah, there's no problem. It's a series of books by Scott Sigler. So he is one of the very first podcasting authors, going back to the early 2000s, where he gave away one of his very first books, or Earth Core. Now, I did not science consult on that. But my questions to him as the author perked his interest in my scientific knowledge. And I've subsequently consulted on his whole pandemic series, which is all too apt and curious, where there is an engineered infection that takes over the population. Also worked on a series of books of Nocturnal, where there is a kind of some monsters in San Francisco. So, and how is that plausible? So yeah, so I just encourage people to go take a look at it. He's definitely a shoot from the hip, raw kind of an author, but it was a fun ride to consult with those. And those are probably the books that I'm most known for. So.
That's fascinating, man. Tell me about where, any of the individuals that listen to this series. Where can people follow you or touch base with you if they wanted to reach out? Where's the best way to find Jeremy?
Yeah, certainly LinkedIn. I'm tapped into that, and I think it's a good nexus for professional development. So you can reach out to me on LinkedIn. It's Jeremy Ellis. You'll see this face in Arizona. You can also email me at, if you're interested from a clinical perspective in my work there, it would be jeremy.ellis@frylabs.com, frylabs.com. But if it's personal, you can email me at my academic email, which is jellis@uci.edu. So those are the
Wonderful. Well, Jeremy, it's been a pleasure. I appreciate the time you spent with us today.
Not a problem. It's great, and I like what you're doing here. It's a lot of fun.